Okay, so what we're going to do in this video here is again focus on alcohols, how alcohols can be used, or the hydroxyl group in the alcohol, how that can be converted into a good leaving group. So we're going to focus on just one aspect of that in this reaction. So we will talk about alcohols as substrates in elimination reactions. And this reaction that we're going to discuss is called a dehydration reaction. So as the name suggests, it is very similar to what happens to us when we are dehydrated. Dehydration would mean the loss of water, okay? So an example for this reaction could be this alcohol. This is a tertiary alcohol. When you react this tertiary alcohol with concentrated sulfuric acid under reflux, we basically reflux a tertiary alcohol with concentrated sulfuric acid under reflux conditions. What you get is elimination. So if you look at that product there, that's not the only product. You're going to get that product plus you're going to get this product. Okay, so you get a more substituted alkene, or the tri-substituted alkene and the di-substituted alkene. And if you look at, let's say, the tri-substituted alkene, and again, since the alcohol is our functional group, this is our alpha carbon. What you've done here is, or what we've gotten here is a double bond between the alpha carbon and beta carbon. Okay, so we have a double bond between alpha and beta. And these two methyl groups, they're both beta prime. And so the other product here is a double bond between the alpha carbon and the beta prime carbon. And what we're losing here is the hydroxyl group and a beta hydrogen. So overall, we're losing a molecule of water. It's a small molecule. So when you start with a substrate and you lose a small molecule from there, this is still an example of an elimination reaction, similar to E2 and E1 reactions. In fact, the mechanism of this reaction is very similar to an E1 reaction. Okay, that is what we would see. There is some similarity with an E1 reaction here. So we lose a molecule of water. That's why it's called a dehydration reaction. Important things that you might want to keep in mind, okay, things that you want to note down are, our reagent here, or our starting material here, is an alcohol, and our products are alkenes. So what a dehydration reaction converts is, what a dehydration reaction does is, it would convert an alcohol into an alkene. The major product here is the more substituted alkene. That's the major product. So similar to E2 reactions when we are doing a Zaitsev elimination. So the major product is the more substituted, the higher substituted alkene. That's our major product. Okay, so what we'll do is we'll look at the mechanism of this reaction now. This reaction is also reversible, but when you do it under reflux with concentrated sulfuric acid, it is more favored in the forward direction. In chapter 8, we're going to see how to do this in the reverse direction. How do you start from an alkene and then make an alcohol? Okay, so we'll talk about that during the mechanism, but the mechanism itself, or the reaction itself, is a reversible reaction. And you get minus water. So there is water as the byproduct. Okay, so that's the reaction. Let's write this down again, and we'll try to write the mechanism. Now the mechanism, we need more space. It's a bigger mechanism, so that's why I'm erasing that space out. So we'll start again here. Okay. So that's our alcohol. And we are reacting this with sulfuric acid and concentrated sulfuric acid, and we are heating this thing overall. Okay. Now, from the reaction itself that we had written down, realize that this is an elimination reaction, which means we need a leaving group in this reaction. But we already talked about this in the previous video. The hydroxyl group in an alcohol is not a good leaving group. It is a very poor leaving group. So what that means is we have to somehow convert this hydroxyl group into a good leaving group. Okay, and that's what the acid is doing here. So sulfuric acid, I'm going to write it out. This is the structure of sulfuric acid, the Lewis structure, okay, of sulfuric acid. And there are protons in sulfuric acid. It's a strong acid. So what happens here is the first step is an acid-base reaction. So the alcohol oxygen uses its lone pairs to get protonated. It would abstract, it would basically deprotonate the sulfuric acid. Electrons here go to that oxygen atom. Okay, these oxygens, they all have two lone pairs to them already. They all have two lone pairs on them. So at the end of this first step, the hydroxyl gets protonated, so we're going to get an OH2 with a positive charge, one lone pair on that oxygen. And along with this, we will have the conjugate base of sulfuric acid. This is an acid-base reaction, very similar to what we did in chapter 3. So we get the conjugate base of sulfuric acid. Oh, sorry, not there. On this, and we have a negative charge over there, okay, on that oxygen. And notice how this conjugate base, okay, this is the conjugate base of sulfuric acid. This conjugate base is resonance stabilized. In fact, it can do a few resonance forms. That's why it's very stable. And if the conjugate base is very stable, then the acid itself should be a strong acid. And the alcohol here is behaving as a base. So this is the conjugate acid of the alcohol, okay? So this is acting as a base. This is our acid. That means this molecule here is our conjugate acid. So now we can do a qualitative analysis of which one is more stable. I would like you to do that. But essentially what it means is this reaction is going in the forward direction because sulfuric acid is a very strong acid. So this reaction is favored in the forward direction. So we make a lot of this conjugate acid of the alcohol, and we make the conjugate base of sulfuric acid. Okay. Now, concentrated sulfuric acid is not 100% sulfuric acid. It's about 98% sulfuric acid. So there is also some water in this reaction. Okay, there is a little bit of water in this reaction. Okay, so we've got this first step done, and it is reversible, but it is favored in the forward direction. Now, the next step, what happens is, now this is where we are going to be changing over to this new way of writing mechanisms where we write things continuously. like in a sequence of events. We're not going to write them stepwise. So far, we've been writing mechanisms as steps, step 1, step 2, step 3. We're done with that because we're basically moving on to like more involved mechanisms, longer mechanisms. So we will not do the step 1, step 2, 3. Although I think this mechanism would be done in about 3 or 4 steps. I don't remember exactly how many, but we'll see. Okay, so this is first step, which is a protonation event. Now that you've made this protonated, you've converted this hydroxyl group into an oxonium ion. That's what we call this, the oxygen with a positive charge. This now is a good leaving group, okay, because imagine this, when this leaves, if this leaves from here, what is it that's going to come out? That's water, oxygen with 2 hydrogens and 2 lone pairs. Water is going to be coming out, which is a very stable molecule, or stable molecule, okay? I think when we say very, we have to compare it to something else. So it's a stable molecule. So we could compare water to the hydroxide that would have come out in the first case. So comparing those two, water is definitely very stable compared to the hydroxide. Okay, so water is produced. And so therefore, this step is favorable. Now we've converted our bad leaving group into a good leaving group. Okay, so the hydroxyl here, this was a bad leaving group, and now we have this oxonium ion, which is a good leaving group. So therefore it leaves, and again, when it leaves, it's going to produce a carbocation plus, it's going to give us a carbocation plus water molecule. Now, we can talk about whether this step is favored in the forward direction or the backward direction, okay? Or is it like equally favored in both directions? Now, when you look at the species, and we're only looking at this step, when you look at the species, your product is a carbocation, water came out, a good leaving group. But in terms of stability overall, if you look at your reactant versus your products, okay, what you notice is in your reactant side, you had this oxonium species, but every element here had a complete octet. Now you've created a carbocation, which is missing, the carbon is missing its octet. You've made water molecule along with that. Okay, so overall, this reaction is not very favored in, it's not very favored in the forward direction. It's actually more favored in the backward direction because you will have a species with complete octets, okay, if you were to evaluate this. Okay, but some of that is going to be formed. You will make some carbocation, and you can favor this process. Okay, so usually the reason why we reflux this is when we are doing this reaction, as we do this reaction in the lab, we try to get rid of that water. We basically distill it out. Okay, so if the water is distilled out, if you remove it from the system overall, then this step is going to be favored in the forward direction by Le Chatelier's principle. So that's how we do it. So now we've got this carbocation formed, and now I think what I forgot to mark here is that's our alpha carbon, this is our alpha carbon, this is our alpha carbon. So similar to an E1 reaction, the carbocation is formed at the alpha carbon. Now that's the alpha carbon, this is our beta carbon, and we have beta prime carbons here. So our beta carbon here has hydrogen, so that's an H beta, and this is an H beta prime. Okay, so in a subsequent step now, realize that I said there is water in the system. Okay, it's not just sulfuric acid, there's water in the system. So this water molecule here, or another water molecule that's in the system, it can come and it can deprotonate that beta hydrogen. Again, this part of the mechanism is similar to an E1 reaction now, okay, because that's what happens in an E1 reaction. You make a carbocation and then the solvent comes and deprotonates the beta hydrogen. So water molecule, it can deprotonate H beta, so it will grab that beta hydrogen. As it does that, electrons here drop into the space between alpha and beta, okay, and you're going to get your product, which is going to be the more substituted alkene, where the double bond is between alpha carbon and beta carbon. Now, the other option is the water molecule comes and deprotonates H beta prime, okay? And then those electrons fall into the space between alpha and beta prime. And so we would get the other product, which is going to be the less substituted alkene, where the double bond is now between alpha and beta prime. Okay, but the outcome of this reaction would be like the major product is the more substituted alkene. So this is our major product. Okay, the stability of the product basically dictates which alkane is formed. And again, since the water molecule is picking up H beta, along with this, you're going to make H3O plus. Sorry, a hydronium ion. You're going to make H3O plus. Now, you could use this conjugate base of sulfuric acid to deprotonate H beta and H beta prime. And in that case, you would make sulfuric acid. You would get your sulfuric acid back. Okay, you would get your sulfuric acid back. So what that means is you only need a tiny bit of acid for doing this reaction, because the acid is used in the first step and it is regenerated in the final step. Okay, so you get your acid back. So this reaction is actually catalytic in acid. So catalytic means you only need small quantities of the acid. Okay, that is enough to do the reaction. And I think what I left out here is in the final step now, you're going from a carbocation, which is a species with incomplete octet, to products where all the octets are complete. So this reaction is actually favored in the forward direction and less favored in the backward direction. But overall, the mechanism is reversible. Every step can go forward and backward, okay, to a decent extent, depending on the step that we're looking at. And that is why what we'll see in chapter 8 is you can start with alkenes and actually go back and make alcohols from the alkene, okay? So like I said, the reaction is overall reversible, but to basically push it to products to get to the alkene, what is done is when this is done in the lab, experimentally when we do it, we try to basically get rid of that water somehow so that you push the equilibrium in favor of the products. So that's how you can get more and more of your product, okay? In fact, this is a lab that I'm not sure where you got to in the 3140 lab, but this is an experiment that you do in the 3140 lab, a dehydration of an alcohol to make alkene. So you may start with cyclohexanol and you make cyclohexene from that reaction, okay? And in that particular reaction, when you do it, you actually distill out your alkene. So it really depends on which of the two, like the alkene or water, which one has a lower boiling point. So the species, the compound which has a lower boiling point, it's easier to distill that out. So when you do that in the lab, the cyclohexene is lower boiling than water, so it's easier to distill that out. So either one, you distill it out to push the reaction forward. Okay, so that's the dehydration reaction. And essentially it makes alkene products follows the same mechanism as an E1 reaction. Okay, let me show you another example. We won't work through mechanisms necessarily, or maybe I can do another mechanism. I think that's fine. Let's do another reaction and actually work through the mechanism. And maybe I'll use the sulfate, the sulfate ion as our, the bisulfate ion as our base in this case. So this second example here, so let's say we have this alcohol here, and you react this with concentrated sulfuric acid. In fact, let's do the example that you actually do in the lab. Let's start with cyclohexanol, and you react this with sulfuric acid with heat, and you make cyclohexene. Okay, and you lose water in this reaction. So plus water comes out in some sense. Okay, plus water is what's coming out. Okay, mechanistically, cyclohexanol is going to react with the sulfuric acid. So there's another variation that I want to show you here, because that's why I thought I'll do the mechanism, is depending on the resource that you're looking at, whether it's the textbook, you're looking at something online, or maybe a different textbook, sometimes you might just see the alcohol written along with H3O plus, like hydronium ion. So H3O plus is water with a proton on it. Okay, that's what a hydronium ion is. Now, a hydronium ion is used to just generally represent acids, because whenever you have an acid, whenever you have an acid, HA, and if there is water present here, okay, if there is water present, then water can act as a base and it can deprotonate your acid, and so that you get hydronium ions plus the conjugate base of the acid. Okay, now the position of this equilibrium depends on the strength of the acid. If your acid is a stronger acid compared to the hydronium ion, then the position of this equilibrium would lie to the products. It would favor the products. Okay, but if HA is a weaker acid than water, then the position of the equilibrium will favor the reactants. Okay, now sulfuric acid is a strong acid compared to a hydronium ion, so the position of this equilibrium actually lies or favors the hydronium ion. So for that reason, often you would see basically H3O plus written. Okay, and if that is the case, so I already showed you how to use the sulfuric acid to do this mechanism, but let's say if you see a hydronium ion written, essentially what you do is you take the lone pair on the oxygen, it's going to go grab that proton from the hydronium ion. So if it is a hydronium ion, please make sure that you write it out as HOH2+, because when it's a mechanism, you want to show all electron pushes that are possible. So the alcohol reacts with the acid, does an acid-base reaction. Electrons here go to the oxygen. So now since I wrote out that oxygen-hydrogen bond, I'm able to show these electron pushes. And on a quiz or an exam, every electron push is going to be some points. Or for every missing electron push, you're going to be missing points, essentially, okay? So make sure that all the detail is included, every electron push. So if a compound has to be written or if a bond has to be shown, drawn out, please draw it out, okay, so that you're able to show all your electron pushes. So the alcohol gets protonated, and what that's going to give me is the protonated hydroxyl group here. This grabbed a proton, so it is protonated, has only one lone pair to it, and plus there is water, which came from this hydronium ion. Okay, I'm going to erase this part here. And again, this reaction would be favored in the forward direction because we're dealing with the strong acid sulfuric acid. The next step, we've made a good leaving group out of that hydroxyl group. So that leaves, this will give us a carbocation plus water molecule, okay? And realize this is our alpha carbon, so the carbocation is at the alpha carbon, or is at the alpha carbon, so this is beta, this is beta. It is basically identical because it's a symmetrical carbocation, so we can use H beta over there. So any of the water molecules that's in the system, it can come deprotonate H beta, electrons from here going to the space between alpha and beta. Okay, again, sorry. This reaction would be mostly favored going back. So when the water molecules deprotonates H beta, it would give us double bond there, alpha and beta, and plus it's going to give us a hydronium ion back. And since we're going from a carbocation to a species with complete octets, this reaction is favored in the forward direction. So again, notice how we started with a hydronium ion and we made a hydronium ion in the end. We regenerated the hydronium ion. So that means you don't need a lot of acid. So this hydronium ion can now go react with another alcohol molecule, do this process again. So it basically keeps going in cycles over and over again. Okay, so you get the hydronium ion. And now in this case, there was only one beta hydrogen, so there is no other product. So we make cyclohexane. Now, whether that's that or this, you can name it. This is cyclohexene, one cyclohexene. Okay, if you want to put the number, but the number is not necessary. It's cyclohexane. So that's the product. So that's the mechanism of a dehydration reaction. There is similarity to an E1 reaction because after we form this protonated hydroxyl group, or once we form the oxonium ion, then we lose the leaving group. So that's the loss of the leaving group, the ionization step in an E1 reaction. And then there is the abstraction of the beta hydrogen to give us the product. Okay, so that's the mechanism of a dehydration reaction. So that's the other strategy to use an alcohol in an elimination reaction. Okay, so we'll stop the video right there. In a subsequent video, now we will talk about how to use this to synthesize molecules, because although these are simple reactions, these are the very first reactions that you're learning, they're very powerful reactions in terms of what can be done with these reactions. So we'll discuss that in a subsequent video. Bye.